Good
Done.
afternoon. Good afternoon. Still time is there to start?
No, sir, we'll start. Just I wanted Prathamesh to start the YouTube link. We'll start immediately, sir.
Done, ma'am. Done.
Yeah.
Yeah, because I will go in between.
Yeah, sure, sir, sure. Another just 2-3 minutes. Once Prathamesh starts the YouTube link, we'll start, sir.
Definitely.
Hello.
Still time is there to start?
No, sir, we'll start. Just I want.
Hello, Megha ma'am.
Hello.
Yes, ma'am. Started. YouTube started.
Okay, sir. Fine. Good afternoon to one and all present for today's session. Today we are going to have a session from Dr. Preeti Anantaraju, and she will be having her talk on unraveling the anti-cancer potentials of natural product. So it's my privilege to welcome Dr. Preeti for today's session. We are happy to have you for today's session, Dr. Preeti ma'am.
Thank you.
Before we start the session, I would like to introduce today's speaker to the audience and the participants that are available. Dr. Preeti, she has received her doctorate in life sciences oncology from JSS Academy of Higher Education and Research in the year 2017. She was the recipient of UGC Maulana Azad National Fellowship from Government of India for pursuing her PhD. She is currently working as a senior project associate in JSS Academy of Higher Education and Research under the DST project. She is in charge of confocal imaging facility established under the DST project at JSS. She has over 10 publications and almost 502 citation and H index of 6. She has presented her work at various national and international conferences, and she has won prizes for the same. She received her industrial training at Gulf Pharmaceutical Industries at Ras Al Khaimah, United Arab Emirates, and she also was working as a junior lecturer at Ras Al Khaimah Medical and Health Science University in Ras Al Khaimah, UAE. She has also been a faculty assistant professor at Pooja Bhagwat Memorial Mahajan PG Center in Mysuru, Karnataka. Dr. Preeti's research is focused on establishing structure anti-canceral activity relationships of certain small molecules and plant metabolites and developing pharmacological agents for the better treatment of cancer. So since all of us have idea that cancer is a very tricky disease, as well as it's very like deadly, and people get scared of cancer, so there are many researchers that are happening in and around cancer, and today's session will help us to focus on few aspects that will be helping us to have anti-cancer potentials or understanding anti-cancer potentials of natural product. So I would request Dr. Preeti ma'am to take over the session, and at the last 10 minutes will be provided for Q&A, so audience can be they can be ready for their questions after the session. Over to you, Preeti ma'am.
Thank you, Megha. Thank you for a very sweet introduction. I hope I'm audible to all of you.
Yes, ma'am, you're audible.
Thank you. So, as Megha has already given a very brief introduction of myself, I'll be just directly talking about the topic. So today I'll be talking, as I understand the audience are more of research scholars, I'll be going through the most basics of cancers and trying to understand what are the molecules that is available from different natural sources and how exactly we establish the anti-cancer activities of these molecules. And then further, as requested by Megha, I would also show you some of the facilities that is available currently at our laboratory, that is Center of Excellence in Molecular Biology and Regenerative Medicine Laboratory, which is established in 2011 at the Department of Biochemistry, JSS Medical College and JSS Academy of Higher Education and Research Center, Mysore. So just moving further with the presentation, as I said, I'll be touching some basic aspects also. I'll be defining what exactly is cancer and what is the current cancer burden and why there is a lot of research that focuses on cancer and the hallmark features of cancer, that what makes cancer so different and so special. And of course, the current treatments that is available and then the anti-cancer agents that is available from natural sources. And I'll also be touching on the drug discovery process, how exactly the drug discovery happens, and what are the different approaches we use, and at last, the facilities that is available at our center. So, according to WHO, yeah, the International Union Against Cancer. According to the International Union Against Cancer, it is cancer is defined as disturbance of growth, right? It is not the normal growth. We all know that cells divide at a particular pace after some times, but when that control of growth is lost and there's excessive proliferation of the cells, which has no apparent relation to the physiological demands, although there is no demand, but the cells keep proliferating, which results in a mass of cell, that is the disturbance in the growth that causes cancer. So if you can see the image there, that is a cancer, it is a tumor which is vascularized, and it exactly looks like a crab, which is nothing but cancer. So that is termed as cancer because of its appearance. And the study of this cancer is called oncology, as we all know. So, what exactly is oncology? It deals with etiology of cancer, the diagnosis of cancer, treatment, prevention, and research. So, why cancer? Because most of us start like as Megha has already mentioned during a talk, people get scared when you hear the word cancer. Right? Cancer is the leading cause of death worldwide, accounting almost 10 million deaths in 2020. Right? So when we look at the distribution of different types of cancers, we have breast cancers, which is leading, which is accounting for almost 2.3 million new cases every year and accounts for about 0.68 million deaths. So it's very alarming rate that we have the increased number of cancer cases. And followed by the breast cancer, we have lung cancer and colorectal cancers. So if you see in both the so we have I request everybody to just mute your microphone so there's no disturbance.
Yeah, ma'am. I have muted. Sorry for the inconvenience.
Okay, thank you. So yeah, as we see the cases, this is actually 2020 Globicon statistics, which is actually even much higher than what is reported in 2023. So why again? Because the number of cancer deaths are also more. Not only the cases are more, it is also that the cancer related deaths are also increasing. Uh, Till now, it was seen that ischemic heart diseases or the CVDs were the highest cause for death. But now we see that the cancer is overtaking the ischemic heart diseases, and the number of cancer related deaths are increasing, which is why there are more research focusing on cancers to have better treatment options to reduce the burden of cancer. So how do we define cancer? There are differences where we can distinguish very easily what a normal cell is and what a cancer cell is, right? But not physiologically, if you see, the appearance is also a little different. The cancer cells are large in number and they have irregular shapes and they divide very quickly. They have large shaped nuclei and the cytoplasmic volume is very small when compared to the nuclei. All these changes with physiological appearances can make it very different from the normal cells. So these are the new hallmarks of cancer. This is defined by, the hallmarks means these are the ones which is upregulated in the cancer, which makes us different from the normal cells. So what is that? They have sustaining proliferative signalings. That means unless there is a signal given to a specific cells, the cells do not grow normally. But in cancers, without this proper signaling, the cells can divide and they do not stop dividing. And they can evade the growth suppressors. So we do have growth suppressors which are being ignored by the cancer cells and they continue to divide. Then we have epigenetic reprogramming. Epigenetics can include the methylation of certain parts of DNA or even the HDAC and HATS the upregulations and downregulation of these enzymes. All these epigenetic reprogramming has happened, which can encourage the cancer cells to grow. And they also avoid destruction and they deregulate the cellular metabolism so that they get excessive energy to multiply, and they resist the cell death, and they evade the senescence. That is, they do not undergo apoptosis, which is a normal physiology, the programmed cell death, and so they just continue to grow. But we do have a lot of cancer treatment, right? Like the chemotherapy, and we have the radiation therapy, surgery, and several pharmacological agents that has been developed. But still, there is a problem, that is side effect. We have a lot of side effect from these um, cancer treatments that is currently available. And the other thing is it's highly expensive. It's not like everybody can afford the cancer treatment, and that adds to the burden of the cancer. And that makes that there is a need for alternative anti-cancer agents that can be developed, which has minimal side effects. So when we think of something that has minimal side effects, our focus just shifts to the natural products, because we all know that natural products are the ones which have less effects, less side effects, and can be potent. So a lot of pharmacological agents are being screened globally, which the source can be from plants, marine organisms, and even from the microorganisms. And these molecules, which are isolated from different natural sources, have proven to have better efficacy and also minimal side effects. And that actually gives a better a platform for the people who are working, and making them realize that there is a need to search such molecules. And these natural products and their structural derivatives with outstanding chemical compositions, especially the secondary metabolites from these plants and microbes, they have lot of cytotoxic effects. They inhibit the proliferation, they inhibit apoptosis, they retard metastasis, and they also suppress the angiogenesis. Angiogenesis, I'm sure everybody is aware of, that is nothing but the blood vessel development, which is happening at a higher rate in the tumor cells. So by suppressing that, we can also actually inhibit the cancers. The other thing that is like most of the antioxidant molecules are actually better anti-proliferative molecules also. So we have molecules like curcumin, resveratrol, then epigenin. All these molecules are which is derived from the plant source. They are all known to have a very good antioxidant activity. So these antioxidant molecules, when they were screened for anti-tumor activity, it was also found that they actually inhibit the cancer cell lines by altering the oxidative stress metabolic pathways, and even some other pathways which include the AKT or the MAP kinase pathways, and they can actually inhibit the proliferation of the cancer cells. This here we can see this is the list of the anti-cancer agents that is found from natural sources, and this is all FDA approved drugs that is currently available for the treatment. From plant source we have vinblastine, vincristine, then Taxol, camptothecin, then doxetol. All these are the plant-derived anti-cancer agents, and even from the microorganisms, we have several molecules that has been identified to have the anti-cancer potentials. And this is a very small number what I have listed. There are many more molecules that is currently available. Yeah, but our focus in our lab where I completed my PhD and I'm currently working is more on the phenolic compounds. And these phenolic compounds, which are secondary metabolites of the plants, and they have lot of diverse structures, and these are found ubiquitously in most of the greens. So these molecules are known to possess both antioxidant as well as anti-cancer activities. So other than the anti-cancer and antioxidant activities, these are also anti-allergic, antiviral, mutagenic. Sometimes even some of them are pro-oxidants. So some of them behave as antioxidants while other behave as pro-oxidants. But both which ways they are able to inhibit the proliferation of the cancer cells. So these are some of the examples of phenolic acids, that is caffeic acid, protoketochoic acid, chlorogenic acid, quercetin, and several other resveratrol. These are some of the polyphenols that is present in the plants and the microbes. So these are some of the papers which is published from our laboratory, which focuses on the phenolic compounds and their role in treatment of cancers. The first one, that is an overview on the role of dietary phenolics. This is a review article which has more than 450 citations till date, which is completely on the role of the phenolic compounds in treating different cancers. And the other two is also like the SAR activities of some of the molecules against the enzyme, that is histone deacetylases, that is usually upregulated in the cancers. Yeah, drug discovery is very time consuming and expensive, right? So when we see this graph, we can see that when the drug discovery is like starting with around 15,000 compounds, but only one drug gets approved. And for this, it takes at least 7 to 8 years of time. So when we see the number of drugs that is being approved than the number of drugs that is being screened, it is very, very less. So we have to be more precise in selecting our starting material from where we are going to screen the our source is, and from how we set up our experiments and how we carry forward our screening processes. And drug discovery is definitely a marathon, because the basic research, that is screening and testing itself, takes almost like 3 to 4 years of time, then goes to the patenting of the molecules, and then you'll have to study its toxic effects, so toxicological studies, and then it enters the preclinical phases and the clinical phases, and by the time we finish and the drug enters the market, minimum of 12 to 15 years is what is required, which is a huge number of time that is being invested in the drug discovery. So how exactly does this drug discovery happen? So there are 3 basic things which we usually do it, that is in silico screening, in vitro screening, and the in vivo screening, and then it comes the clinical trials. So I'll be explaining about what exactly is in silico, in vitro, and in vivo in terms with respect to the anti-cancer drug development. Yeah, this is nothing but in silico work, that is using the system, that is the computer, the bioinformatics. We are going to we are going to identify certain targets, then we validate those targets, and we use the molecules. When we know the structure of the molecules, we use certain targets and try and see whether this drug is interacting with our target, so that whether this interaction can have a positive effect or negative effect can be screened later. So the interaction of the molecules with certain structural aspects is very important, which can be screened using in silico work. And the advantage of having an in silico work is it is a high throughput screening. So you can screen a lot of molecules, that is like thousands of molecules at a time, which will not be possible if you start doing an in vitro assay. So initially, when we are going to screen, we can use the in silico approach, that is the computer-aided discovery. And there are several softwares that is available, and this is one of the software which we have used during our study. This is called as Discovery Studio, and here we have taken HDAC as the target, and we have screened almost 20 molecules for the interaction between this HDAC. And in that, we have found that 2 molecules are very potent and they have a better interaction with these HDAC molecules. And then we have taken only those 2 molecules for the in vitro assays. So it becomes easier to carry out the in vitro assay, because taking all the 15 molecules or 20 molecules and screening them in vitro is very challenging, time consuming, and of course, very, very expensive. So in that case, the in silico approach is going to definitely help the scholars who can screen these molecules at a quicker pace and then decide on to which molecule to move forward with. So once we have decided that among 15 or 20 molecules which we are screening, 1 or 2 molecules are showing better activity, we go for in vitro testing. So in vitro testing is all cell-based assays for developing the anti-cancer activities. So we need the cells to be procured either from the globally we have ATCC, that is American Type Culture Collection, which is there in the USA, which provides the cell lines. And in India, we have NCCS, that is National Center for Cell Sciences, Pune, which provides the cell lines for the in vitro assays. So these are some of the examples of cell lines. This is HeLa cell line, which is the first two images. And this HeLa cell lines is nothing but the cervical cancer cell line that has been established. And this is one of the very aggressive cell lines and commonly used cell to test different anti-cancer agents. And the other one here on the right-hand side is HCT116, which is a colon cancer cell line. And these cell lines, the advantage with having cell line is we can grow them in vitro and we can test test the agents or these drugs what is of our interest on these cell lines. So how do we test them is like we do have the cell facility, the cell culture facility, which has the like this is how a cell culture facility look like. We have the biosafety cabinets and then the CO2 incubator, which is maintained at 37 degrees with 5% carbon dioxide and relative humidity of 97%. Then we use certain media and the fetal bovine serum. This is FBS is fetal bovine serum, which is used at 10%, which provides the microenvironment for these cell lines to grow. And then we have this glutamax, and which is the source of the amino acids. And then we have pen strip, an antibiotic to minimize the growth of microbial contaminants. With this media, we can usually grow the cells and we can use different assays to establish the anti-cancer activity. What are those assays? There are several assays which we can use. So the first and foremost assay which we do is the cell viability assay. So we are going to check that how much of cells are alive or dead when we use a compound for treatment, right? So we do either MTT assay or SRB assay. There are still more other assays like MTS assay and others. But usually the most widely used ones are MTT assay and SRB assay. And then we do cell proliferation assay. What is the rate of proliferation when compared to your controls and the treated, whether the proliferation is reduced or not. If the proliferation is reduced by the drug of our interest, it is like more promising that it is actually inhibiting the cancer cell growth, right? Then apoptosis assay. Yes, apoptosis, as I told, cancer cells do not undergo apoptosis very easily. They have bypassed the mechanism. So sometimes these anti-cancer drugs, they can activate certain cascades of apoptosis, including the caspase activity, especially the caspase 3, caspase 8 are some of the caspases which are usually activated by these anti-cancer agents, by which the apoptosis is also enhanced and they undergo cell death. Then we also check for cell migration assays, where we can actually see if these molecules can inhibit the metastasis. The cell migration assay is a model to study the metastasis of cancer cells on if the drugs can inhibit the metastasis. Then, of course, we have cell cycle analysis, where we are going to check whether these molecules can inhibit the cell cycles. And angiogenesis assay, of course, as I said, this angiogenesis, this new formation, blood vessels that are formed, if we can inhibit the formation of new blood vessels, we can actually kill the cancer cells. And after doing this, we also look at some mechanism of action, that is either by doing PCR, where we look into the mRNA level, and of course the Western blot, the protein expressions can also be studied, so that it becomes very clear by which pathway is our anti-cancer molecule actually inhibiting the proliferation or killing the cancer cells. So once we find that these molecules are good, that it is killing the cancer cells, the next available option is we go for the in vivo studies, because in vitro studies, we will not understand about the bioavailability of the molecules. So we are not sure if we give some particular dose, whether all of that dose can be utilized when it is consumed as a whole drug. So that's why the in vivo models are used. And usually for the cancer, there are options like we use usually the Swiss albino mice. And we also use the immunocompromised mice, that is also called as xenografts. It is very rare that we use C57BL par 6, that is a black mice. Black mice is usually used for diabetes studies, but most of us especially use the Swiss albino mice. And xenografts, that is the immunocompromised mice, it is challenging to handle them, but it is a very good model to study the cancers. So in the EAC, that in the Swiss albino mice, we do induce two types of tumors, that is either the liquid tumor model or the solid tumor model. So in the liquid tumor, it is the same cells, that is Ehrlich's ascites carcinoma, which we call it as EAC cells. These are types of breast cancer cells that is originated in the mouse. So if we inject these cells intraperitoneally to the mice, we get the liquid tumor that is accumulated. The first image, what we can see here, this is the control mice, and this is the liquid tumor bearing mice. We can see how much the liquid is accumulated, right? So once it accumulates, then we start giving our treatment and see whether this EAC reduction is happening or not. The same way, when the same cells, that is EAC cells, are subcutaneously injected to the mice, we usually do the subcutaneous injection near the thigh region of the mice, and that develops into solid tumor models. And these solid tumor models can also be used for studying the anti-cancer effect of the molecules. And now these drugs of our choice can be either given orally or can also be given through IV or IP. Usually, if it is a plant extract or a microbial extract, we prefer to give it orally because that is the mode how, if it is converted into a drug, it is usually taken up. So oral is preferred when we are doing such experiments. And the other model is the immunocompromised mice. Again, in this, we have two types, that is the cell line derived xenografts models or the patient models. So this is not PDX, I'm very sorry for that. It is CDX, that is cell line derived xenografts. So what we do, that is human cell line that is already growing in the lab can be injected to the mice, which is immunodeficient mice. So the immune system is so low that when we transplant these cells, it grows into a tumor and this mice can be used for the study. And the current model that is available is the patient derived xenografts model, where the tumor directly from the patient is taken, it is cultured in the laboratory and then injected to the mice. This is the level of personalized medicine that is happening currently in the cancer treatment, where the patient-derived xenograft models have been used to study whether the drug of other choice is having any impact on the cancer of that particular patient. So this is like currently happening in India. Yes, in NCBS also we have some of the patient-derived xenograft models that has been developed, and they are using currently these models. So when we have an experimental, this is just an example of how exactly the animal work is done. We do take the animals and acclimatize them for around 7 days, and then we inoculate the cells, that is either the liquid tumor or the solid tumor on day 1, and wait for 7 days for the tumor to develop and then start the treatment. And we continue the treatment up to 21 days. And this time is not specified as such, but this was one of the experiment designs what we have used. So I've just quoted that example. So after 21 days, the mice is been actually, it is sacrificed and on sampling is done. And while sampling, we usually collect the serum samples, tumor tissues, and certain vital organs, that is liver and pancreas. So from the serum, we do a lot of biochemical parameters. Then from the tumor tissues, we do RNA and protein expression studies. So protein expression studies is either done by IHC or even by the Western blottings. Then RNA by the qPCR method, we usually see the expression. And then we also collect these vital organs, that is liver and pancreas, and check for H&E staining just to make sure that these molecules, do they exert any toxicity on the cells or not? Because we don't want our treatment, the drug, to have certain side effects or maximum toxicity for the normal cells, right? So that is why we do the H&E staining and confirm whether our molecule is having any toxicity or not. So this was the 3 different approaches, what I just briefly explained, which is in silico, in vitro, and in vivo methods of approaches for the anti-cancer treatment. Any questions so far?
I guess, ma'am, it will be better if we have the questions at the end so that you'll have continuity.
Right. So with this, I'll be actually ending the first part of my talk where I have spoken about what are the different approaches of anti-cancer drug treatment. So now I'll be showing the facilities that is currently available at CMR so that it is useful for the research scholars who are planning to do their work in this area of interest. Very sorry. Yeah. So this is the CMR facility, which was inaugurated at 2011. Then we right. So this is the lab. Here you can see this is CMR lab, and this is our cell culture lab, which is currently available for the anti-cancer drug screening. And this is the areas of focus at CMR. We do a lot of cancer biology studies, fatty liver disease studies, sepsis, cardiovascular diseases, COPD, and diabetes. But a lot of work is happening on cancer biology, where we have three Ramalinga Swamy fellows from US have come back and they have started their career in CMR. And we do have sepsis, which is a new area of our interest, which is headed by Dr. Akila Prashant. And then, of course, cancer biology, there is a lot of work going on under one of the very senior professor, Dr. Subbarao. And COPD and cardiovascular diseases is also being taken up by one of our professors, that is Dr. Rajesh, who is all doing different aspects of their research. And there are almost currently, I think, around 25 to 30 research scholars at CMR who is currently pursuing their PhD. So these are some of the facilities that is available. We do have a world-class facility for cell culture is what I would like to say. We have almost 4 biosafety cabinets and 3 incubators where we have dedicated spaces for different cell lines to grow. And we do have this fluorescence microscopes and the multimode plate readers and the PCR machines, qPCRs. And then we have these gel dock systems that is used for the protein expression studies. We have nucleocounters for analyzing the cell cycles. And of course, the new addition is the FACS lyrics BD from BD Sciences, which is also the flow cytometer that is used for different aspects of cancer biological studies. And then we have this Telesar 5 confocal microscope, which is again procured from DST PERS project, which is currently a new addition to the laboratory. And then we do have certain instruments that is required for phytochemical screening. And one such one is the rotary evaporator, what we have. Excuse me. And then we have this liquid chromatography and a high resolution mass spectrometer from SciEx, which we are using currently for the phytochemical analysis and characterization. And we also do some of the metabolomes and the proteomic studies using this LCMS. We also have a HPLC system and zeta sizes for the nanotechnology studies. In addition, we also have a very good Center for Experimental Pharmacology and Toxicology at JSS Aher, that is a central animal facility. And this instrument on the right is the Wilbur Newton live animal imaging. I would proudly say that this is the first instrument in India which is present at JSS Aher, which facilitates the imaging of the live animals. So without sacrificing the mice, we can study some of the aspects using this instrument. So yes, with this, these are different funding agencies from where we have a lot of funds which is coming into the CMR. We have DST PERS project, which is almost of 9 to 10 crore project. And then we have the DBT Builder project, again, which is a very huge project. We have again India Alliance from DBT. Then we have also the VGST and ICMR. Recently, the center CMR laboratory is also recognized as ICMR Center of Collaborating Excellence, which is an added feather to the lab. This is our campus. This is the JSS Medical College, which is present at Mysuru, where our lab is situated at Department of Biochemistry. This will be the global campus that is upcoming very shortly, like maybe another 1 or 2 years, we will have this global campus. And the other advantage for having research at JSS would be like we also have the JSS hospital, which is 1,600 bedded hospital. So there are ample of cancer samples also available from the hospital, since we have a very good setup of oncology department who are also very much interested in research. They do collaborate with the basic researchers and they provide a lot of samples. So it becomes easy for us to have these samples done and carry out our research. So of course, talking about Mysuru, I was not able to withhold without showing you some of the important places at Mysore, right? Mysore Palace is very famous, and then we have the Keras Dam and the Philomena's Cathedral, and of course the Chamundi Hills, and yeah, Mysore Dasara, the most famous world famous Mysore Dasara, which is happening. And we are very happy that our institution is present in such a beautiful city, which nurtures a lot of research and inspires the students to do a lot of research. With this, I would like to end my talk. Thank you for your patience hearing. It was all a very basic study and outline what I have given, since it was requested that it is most of the research scholars who are there. So I would be happy if I have any questions to be answered.
The session is open for question and answers. The participants can unmute yourself and raise your questions.
Yeah, ma'am. Good evening. This is Dr. Amit Patnaik from NIST Barampur. Am I
Yes.
audible?
Yes, please tell me.
Yeah, Dr. Preeti, ma'am. I want to know ki actually my PhD is on the cancer cell lines, but the problem is ki when we are having some very good result with the cancer cell lines and mice and all the rest models, but when we translate that to the human cells or biopsy samples or directly to the live human subject,
Right.
so that type of result we are not getting with that. So the normal cells are also affected and it will get dying for
Yes.
that.
Yes, so see, that is one of the most critical thing when we are screening any drug for an anti-cancer activity. It is mandatory to also check it on the normal cells, right? So if normal cells are available, like in our lab, we do have the normal keratinocytes and normal lung cells. So we do test it on them, because the first and foremost criteria for an anti-cancer molecule
Ma'am,
should be like, yeah, please.
yeah. And another problem with when we are going for crude extract, say some plant, we are taking the crude extract with some solvents,
Yes.
and another thing is in crude extract there is a mixture of bioactive compounds, right?
Yes.
And when we are going for and searching for a single bioactive compound from that, the mixture of the say 100, 200 bioactive compounds, that is another tedious work to do.
Yes.
So, what sort of solution you are giving in that?
So usually we do is bioactivity guided fractionation can be followed, where you have different fractions and you select one of your fraction which has highest activity, subject that into the chromatography, either the gel chromatography like the size exclusion chromatography or affinity chromatography, because you do have a rough idea on what kind of molecules you are looking at.
Yeah,
So
yeah.
based on that, you need to isolate your molecules. Of course, challenging. It's very, very challenging, because isolation and characterization of a molecule itself can take almost like 4 to 5 years of your time. So I would suggest that is a very challenging task.
And one more thing I want to add here, ma'am, if we are going for a funding agency for this extract preparation and all that, getting a funding for that is very challenging for us.
Right.
So,
Because yeah, that is because we are not telling them that we are going to isolate one particular molecule which is very, very active, because a lot of people are working on the fractionation and isolation of molecules. So the yield is like the production of novel molecule is something is what the funding agencies are looking forward. So when we are saying that it is just a crude extract, I don't think so. The funding agencies are really interested in funding. So we need to be more precise on telling, like we'll be like at least, purifying the crude extract for some extent and trying to identify the novel molecule that is responsible for the activity that could maybe attract more funds from the funding agencies.
Yes, ma'am. Yeah. Thank you.
Preeti ma'am, I have a question.
Yes, please.
Like when you started your talk, you told that the compound that you isolated from the plant was a phenolic compound, right? So even there are other phytochemicals that are present in the plant, like carbohydrates, lipids, and the nitrogen containing. Is any other work related to this anti-cancer property of these compounds done?
Yes, it is not, my work was not isolation of molecules. What we have
Okay.
taken is the natural, like the synthetic compounds that is available. But we do lot of extractions in our laboratory. So your question is how I could isolate, when we could isolate certain class of molecules, right?
No. My question is, apart from phenolic compounds that are actually used for checking the anti-cancer property, there are other phytochemicals also in the plant. So, is any work related to anti-cancer property of those phytochemicals done?
No, our lab majorly focuses on phenolic acids, and that is why we use usually 70% ethanolic extraction. So when
Okay.
we use 70% ethanol for as a solvent for extraction, it is usually the phenolic compounds which come into the extract, where we
Okay.
exclude all the others. So we have characterized these phenolic acids using the LCMS.
Okay.
So then we'll make sure that only these like it is obviously that we do have other molecules other than phenolic acids. It's not like very pure phenolic compounds we have.
Okay, ma'am.
Right. So when we do the like the isolation using the gel chromatography or the affinity chromatography, then we are only sure that we have like one or two molecules.
Okay,
It
ma'am.
could be because of the property could be not because of one single molecule. It could be because of many different molecules.
Many different molecules. Yeah, true. Any more questions from the participants?
Yeah, Megha,
Yes,
can I
yes,
ask
ma'am.
my
I have a
question?
question.
Yeah.
Hello, ma'am. Your presentation is very interesting. This is Swati from Hyderabad.
Yeah, thank you.
Ma'am, can you hear me, ma'am?
Yes, Swati, please tell me.
Yeah. Ma'am, actually, in your presentation, you have shown plant extract and the nanoparticles, right?
Yeah.
In one of these slides. So, is there any one novel nanoparticle that is proven to be anti-cancerous, ma'am?
Yes, we do. Like when you find out that there is a molecule which is potent, we try to make it as a nanoparticle because, the nanoparticles can be a better delivery system. It can
Okay.
also have the targeted therapeutics. There are certain molecules, but currently in our lab, we do not work on nanotechnology.
Okay.
We have just started the work with nanotechnology. We do have the facilities for having the nanotechnological type of works, but currently the expertise is available. So we are
Okay.
also
Because when we go through with the research papers, ma'am, in research papers, actually with silver nanoparticles, zinc nanoparticles, and iron nanoparticles, there is a lot of work going
use
on
lot of
related to
all heavy metals are also used, like cobalt is used, copper is used, zinc is
Yes,
used,
ma'am.
silver
Yes.
is used, and
Yeah.
they do also some green chemistry synthesis of nanoparticles.
Yes, ma'am. Yeah.
So it all depends on your particular compound and how the metal ion or whatever you're selecting for the nanoparticle, their interactions, and also probably. I'm not
Yes,
an
ma'am.
expert of that field to
Okay.
talk about that.
Yeah. I have another question, ma'am.
Yeah.
Like, for the research scholars, can we just simply do our research till the in silico and then the lab secretion? That's it, without going to the in vivo.
That is up to you because it depends on how well funded your project is, because of course in vivo work has lot of expenses.
Yeah, it's very expensive.
Very
So
expensive. But I would suggest, like, if in our lab it is usually that we have to do the in vivo work, because otherwise, the acceptance of papers are also very challenging.
yeah, yes, ma'am.
Yeah, so, and the complete understanding of a drug, how it behaves, is only when you carry it in the animal studies. Because your
Yeah.
molecule can be an excellent drug when you're doing it in vitro, but there are cases that it has failed when you have taken into the animal studies.
Yes.
So, animal
Because
studies would actually be better if it is conducted, especially when we are doing such, like, anti-cancer drug screening. Maybe if not two or three molecules, at least one single, the most potent molecule, whatever you've identified, it is always better you do it in the animals.
because in vivo studies itself are very expensive, and then at the college level where we don't have any funding, then for the research scholars, so I think it's very difficult now.
Yes, I
Yeah.
do understand, but yeah,
Yeah.
it's the way for. But especially if you're working with plant molecules, where you have not done a very particular isolation, it's just a crude extraction you have done. So it is very important that you establish the mechanism.
Yes, ma'am.
So with
Even
that,
the microbial synthesis of certain anti-cancerous products
yes.
is very interesting, but then even that isolation is, I think it's very difficult, na, ma'am.
Yes, see, there are two approaches which you can do. One, when you identify that there is one single novel molecule in your extract, but you are unable to isolate that in a large quantity, because for which you are, like, you need in a lot of quantity for in vitro and in vivo work. But if
Yeah.
that molecule is already available in the market, you can purchase the synthetic version of the same molecule and try the efficacy of that molecule.
Okay.
Right? Because isolation itself is going to take you like 3 to 4 years of time, and then doing the animal work. Because I would suggest animal work should be like, at least you should keep 1 year of your time for animal work.
Okay.
Although the study is for just 1 month or 2 months, but the process involved is too much. So you need to spare at least 6 months, minimum of 6 months for your animal work.
Okay. Thank you so much, ma'am.
Thank you.
Hello.
Yes.
Yes, ma'am.
Hello.
Yeah, please ask your
मैम
question.
Yeah, please.
मैं गौतमी ठाणेकर बोल रही हूँ मैम, शिवाजी यूनिवर्सिटी से। मेरा ऐसा क्वेश्चन था कि जैसे हम लैब में एंटी माइक्रोबियल एक्टिविटी जैसे देख सकते हैं, वैसे ये एंटी कैंसरस एक्टिविटी नहीं देख सकते, क्योंकि और इसके लिए जो प्राइसिंग है वो भी ज्यादा है। तो इसके लिए हम कुछ कर सकते हैं कि ये हमें छोटे लैब में भी कर पाए?
We can do in vitro screening of the anti-cancer activity. That's what I showed, that there are cell-based assays where we can do. But of course, when the cost, you cannot compare the cost of a microbiology lab to an animal cell culture lab.
हाँ,
It's very, very
वही
expensive.
प्रॉब्लम होता है।
Yeah, it is very, very expensive, because
Yes, yes, ma'am.
the media and the supplements that is required for growing these cells, they are very, very expensive.
Okay, ma'am.
So just if you have seen the slides here, the FBS bottle, what I have shown you, it's just like,
हाँ हाँ
this FBS bottle, this single bottle is around 50, right now, I think 18 to 19,000 rupees, a single bottle. So it is expensive. Yeah. So cell culture work, has to go through a particular standards, so it cannot be bypassed, and there's no alternative for that.
Any questions? Participants, if you have any questions, please unmute yourself. Actually, Preeti ma'am, the topic of cancer and anti-cancer agent is an unending process where you keep on reading things and you keep on having queries. So actually, I have multiple questions, but putting out the questions at this time will not be right. So I'll reach to you personally for the questions. So I
Okay.
guess today it was really a very knowledgeable session. the repository of the information about the cancer and about the anti-cancer agents and its effect on the cell lines as well as the live creatures. The way you have shared your knowledge and your work on anti-cancer products have really enlightened our knowledge about anti-cancer and put a new light on the area where still more work can be done and few more things can be discovered. So I'm really thankful for your time, and it's our pleasure to have you for today's session. Thank you, Preeti ma'am.
Thank you, Megha. Whatever I have covered is the most basic thing, I suppose, because there are a lot more, as you said. We are advanced in different ways. There are even 3D organoid cultures that is growing, but I have just covered the most basic aspects.
Yeah, ma'am.
It's a pleasure talking, and I would like to thank Megha and also the All India President for Microbiological Society of India, Dr. Arvind, and of course Megha, who was in contact with me, and she has asked me to deliver a talk. Thank you, Megha, for your opportunity.
It's a pleasure, ma'am, to have you. And instead, I'll request you, if you have more and more knowledge about this phytochemicals and the anti-cancer agents, we'll be happy to listen to your talk at least once in 15 days.
Oh, why not? Sure.
Sure. Yeah, ma'am.
Yeah. All right.
Yeah. Yeah, even I would like to thank Dr. Deshmukh sir, who has always been support for conducting such webinars and series of lectures on Sundays and Saturdays, which is actually helping all the scholars to get aware of the stuff that is going in the society and how the research is moving ahead in life sciences. And last but not the least, I would like to thank Prathamesh and all the participants who have took out time from their busy schedule, though it's a weekend, and all of you have shown the enthusiasm for attending the session and making this session very happy and successful. So I would like to thank all of you again. Thank you, Preeti ma'am, for your valuable time. Thanks a lot.
Thank you, Megha. Any questions? Anytime you people can reach me out through Megha, I suppose.
Yeah, ma'am. Sure. I'll be in touch with Preeti ma'am. So any participants who want to get their queries solved can contact me through the group Microbiology Society, and just post me your questions so that I can get the answers through ma'am.
Sure.
Oh, okay, ma'am. So shall we end the session, ma'am?
Yeah, sure.
Sure. Thank you. Thanks a lot. Thank you, all the participants.